Hello, future pharmacists. Today, we're diving into high-performance liquid chromatography, or HPLC, a powerful tool for analyzing medicines. HPLC acts like a molecular detective, ensuring our drugs are pure and safe. It separates, identifies, and measures each component in a mixture. Think of sorting candies by color, but with invisible molecules. In pharmaceuticals, HPLC checks if the right amount of active drug is present and detects any impurities. The process involves pushing a liquid sample through a tube packed with a special material, the stationary phase. The liquid that moves is the mobile phase. Each compound in the sample interacts differently with the stationary phase. Some stick and move slowly, others zip through. This difference in speed separates the compounds. By the end, each component exits the tube one by one, perfectly sorted. It's like a race where some runners get stuck and finish last, while others sprint ahead. This principle of separation is the foundation of chromatography. HPLC is used everywhere: hospitals, food science, environmental testing, and most importantly, pharmaceutical quality control. It's essential for ensuring every medicine is safe and effective. Let's see how it works. Let's break down the HPLC machine. First, solvent reservoirs hold the ultra-pure mobile phase, the liquid that carries our sample. The pump pushes this liquid through the system at high pressure, ensuring a steady flow. Next, the injector introduces a tiny, precise amount of sample into the moving stream. The column, packed with stationary phase material, is where separation happens. As the sample travels through, compounds separate based on how much they interact with the stationary phase. The detector then identifies each compound as it exits the column, sending signals to a computer. The computer creates a chromatogram, a graph showing each separated compound. Every part of the HPLC system works together for accurate analysis. This teamwork is what makes HPLC so powerful. Now let's follow a sample's journey. Here's how a sample moves through HPLC, step by step. First, the injector loads a tiny amount of prepared sample into the mobile phase. The pump pushes this mixture toward the column at a constant rate. Inside the column, compounds separate. Some stick to the stationary phase and move slowly, others move quickly. As each compound exits, the detector measures it and sends data to a computer. The computer generates a chromatogram. Each peak is a different compound. The time a peak appears helps identify the compound. The peak's size shows how much is present. This process gives us a clear, detailed analysis of our sample. HPLC's precision is why it's trusted in pharmaceutical labs. Every step, from injection to detection, is crucial for reliable results. That's the journey of a sample through HPLC. Next, let's look at the types of HPLC. Choosing the right stationary and mobile phases is key in HPLC. The two main types are normal phase and reverse phase HPLC. In normal phase, the stationary phase is polar, and the mobile phase is non-polar. Polar compounds stick to the column and move slowly. Non-polar compounds pass through quickly. In reverse phase, the most common type, the stationary phase is non-polar and the mobile phase is polar. Here, non-polar compounds stick and move slowly, while polar compounds exit first. Adjusting the mobile phase changes how fast compounds move, allowing precise control over separation. Reverse phase is popular because it works well for most drugs and is easy to use. The choice between normal and reverse phase depends on the properties of the compounds being analyzed. This flexibility makes HPLC incredibly versatile. By selecting the right mode, we can separate almost any mixture. That's how HPLC adapts to different analytical challenges. Now, let's see how it keeps our medicines safe. HPLC is vital for pharmacy and beyond. Besides normal and reverse phase, there are types like ion exchange, separates by charge, and size exclusion, separates by size. HPLC is used for purity testing, quality control, and stability testing, ensuring medicines are safe and effective. It checks drug concentrations, finds impurities, and helps determine shelf life. In pharmacokinetics, HPLC tracks how drugs move through the body. It's also used in forensics and clinical monitoring. HPLC is the gatekeeper of medicine quality and safety. Understanding HPLC is essential for every pharmacist. It's a cornerstone of modern pharmacy and public health. Science is cool, and HPLC proves it.